Hi everyone, this is Yueting Li from Stanford University. Today I'm going to present our paper named Joint Graph Convolution for Analyzing Brain Structural and Functional Connectome. First of all, please let me introduce some background information to make it more clear. The human connectome refers to the concept of describing the brain's structural and functional organization as large-scale complex brain network. A fundamental problem for system neuroscience is determining the best way to relate structural and functional networks quantified by diffusion tensor imaging and the resting state functional MRI. As one of the state-of-the-art approaches for network analysis, graph convolutional networks has been separately used to analyze structural and functional networks, but have not been applied to explore inter-network relationships. To explore that, in our work, we have two goals. Our first goal is to couple brain structural and functional networks of an individual into a joint graph by adding inter-network edges between corresponding brain regions. Secondly, we want to pass this joint graph as the input to a single GCN and accurately predict brain-related attributes and inter-network coupling strengths. Now, let me briefly introduce the method we are using. From the dataset, we get diffusion tensor imaging data as well as resting state fMRI data. The DTI represents structural connectivity of the brain, and the RS fMRI represents the functional connectivity of the brain. Then, we join the two networks together using neural coupling strength vector C. The internetwork edges vector C represents the coupling strength between the SC and FC for each brain region. By doing so, we can get a 2n by 2n adjacent matrix A of SC to FC joint graph. This joint graph contains information for both structural connectivity as well as functional connectivity. It also contains the information of the coupling strength, which is the neural parameter vector C. Then we feed this graph into a single graph convolutional neural network and do some pooling operation. After that, we feed the output to a MLP and get the final prediction result. For example, the Y prediction here can be the age of a subject. The dataset we use is from the public dataset of National Consortium on ACO and Neural Development in Adolescence. We used 1979 pairs of RS fMRI and DTI scans from 328 males and 334 females who have no to low alcohol intake, and their ages are from 12 to 21 years old at baseline visits. We have two tasks, including sex classification and age regression. And here are our final results. We compared our model with some basic models such as MLP, GCN, and SVM, as well as some state-of-the-art models which are also listed here. We compared all those models in multiple modalities, include FC only, SC only, and multimodal. Our model gets the best in accuracy, AUC, Pearson correlation, and second best in the MAE metric. The result indicates models that use multimodal networks has better performance. This phenomenon indicates SC and FC networks contain complementary information about the brain connectome that is useful for identifying sex and age differences. The heat map shows the coupling strengths of bilateral brain regions near in the age and sex prediction tasks. The neuron coupling strengths highly coincident between the two prediction tasks, indicating our learning framework can extract intrinsic SC to SC relationships in the adolescent brain. And here are the references we mentioned in previous slides. And that's all. We'll come to our poster session, which is from 3 to 4 p.m. on September 21 Singapore time, and thanks for NIH and Stanford HAI GCP for their kind support.